Marisa. I'm a postdoc currently at NASA Ames Research Center, and I work on microbial sources of hopanoid lipid biomarkers in different hot spring environments. Today I'm going to be excitedly sharing with you some new potential microbial sources of 2 and 3 methylhopanoids that we were able to find in some of our anoxic, acidic, and sulfidic hydrothermal springs. I want to acknowledge my funding sources, which is ORAU NPP program, National Park Service, NASA, and finally Stanford University, because some of this research was done back in my PhD as well. You might be wondering, if you are an astronomer or anyone but a lipid chemist, what are hopanoids and what do they teach us in modern environments? Why should we care? So hopanoids are cyclic triterpenoid membrane lipids, so they're complex. They're a little bit different from some of the phospholipid membranes we've been seeing today. Hopanoids are some of the most abundant bacterial lipid biomarkers on Earth, so they're very worthy of studying. They are important geologically. They can withstand the lithification process and all of the diagenetic forces that happen when you are becoming a fossil. And so they're actually one of the few really strong sources through deep time of microbial inputs into the rock record. And they can be detected billions of years after deposition. Finally, in modern acidic springs, we observe methylated hopanoids. In this work, we in modern acidic springs observed methylated hopanoids. And we found these in an anoxic environment where no cyanobacteria and no methanotrophs were present. These are kind of the typified known producers of these kinds of biomarkers. So it was exciting to find them at all. And the work that once we found those methylated hopanoids and the follow-up work was able to lead to discovery of some new potential source organisms for these lipid biomarkers, which if you are a biomarker nerd is always exciting and it's ever expanding. So this kind of new evidence of source organisms is important because these kind of anoxic environments and knowing about them and the microbes that live in them today can have implications for us to reinterpret the sedimentary rock record on Earth, but also potentially on Mars or on other planets where we have a sedimentary rock record. So my samples were taken in 2 sites. One is in Yellowstone National Park and one is in Lassen Volcanic National Park. In both springs, you can see the mat is kind of similar. There's a purple layer, there's like a acidic algae layer, and then there's this white flocculent elemental sulfur on top. That's from the actual hydrothermal system itself. So what I did is I go into these springs and I take samples and I go back to my lab and I extract for both DNA and for lipids. And then from there I analyze them. So I found a bunch of different hopanoids. So I did a LCMS analysis on them. And so I was able to actually look at the functionalized extended hopanoids. And so as you can see, there's a lot of colors on that graph. Every color is a different type of hopanoid that I was able to find in my springs. The green overlay on some of them and the pink overlays on some of them indicate a 2-methyl for green or a 3-methyl for pink. So we found a huge diversity of methylated hopanoid compounds. Over to the far right, you can see my controls. We had pretty low diversity of methylated compounds, so it was really interesting in the environmental samples to see so many different kinds, which is what sent us on our DNA work. So the first thing that we did was do a 16S amplicon analysis of our extracted DNA. In that, we didn't see any obvious sources or classical sources of those methylated hopanoids, which was pretty exciting. So that then led us to go back and then do some metagenomics. And then when we were able to analyze that metagenomic data, we saw many potential sources of novel 2-methylhopanoid producers, mostly anaerobic water and soil-dwelling microbes, but way more sources than are currently understood. And this is just from looking in 2 hot springs. So a case for doing field work to expand our knowledge in the lab. We also saw a ton of potential novel sources of 3-methylhopanoids. These were mostly anaerobic and acidic environment dwellers that we found them tagging against. And Yeah, it was pretty exciting. So if you want to know more about either lipids or DNA analyses or microbial ecology of these acidic springs, please come check out my poster later and find me. I'll be at board 22 at this next session. And also, because I am in a room full of early career researchers, I coordinate a division seminar at NASA Ames, and we would love to hear you guys give talks. So if you would like to give a talk at NASA Ames at any point, please come find me at my poster, and I'd love to write your information down, and we would love to have you give a talk. So yeah, we're friendly.
Wow.